I will say that I'll try to call someone and see if they can't fix that for our last few days of class anyway. Let someone know. A few announcements today before we get started. Don't forget today is DNA Day. Check out, yeah, I know. DNA gets a whole day. Check out some of the events that are happening at Campus Center. I think they're happening all day. Stop in and see what's going on. Celebrate DNA. Hopefully you have a greater appreciation for now. Looking ahead for this course, we only have 5 days of lectures left, including today. I know. Aww. They're going to be a little bit sad. I'm looking ahead. So this class is the final is scheduled for the last day of finals. Not the best timing, Friday the 13th, 7:30 to 9:30 a.m. Remember that the final is cumulative. So anything we've covered throughout the semester is fair game, including this last section. Remember that the final is worth 25% of your grade. You cannot drop the final. So you have to be here. You have to take that exam. And it's a fairly significant portion of your grade. So you want to take it seriously. You have lots of time to study though. So it shouldn't be a problem, right? Also, because it's 25% of your grade is the final, it's cumulative, expect the exam to be about twice as long as normal. So instead of 125 points, it will be roughly 250 points. All right? I'm hearing a lot of questions. I'm letting you know now so you're prepared for the exam. All right, generally I haven't completely made up my mind yet about what the breakdown is going to be. There'll be slightly more questions on the new material relative to the old, but I haven't decided exactly what the breakdown is, whether it's 50-50. I haven't made the exam yet, so I don't know. Okay? To start thinking about that exam, I will get a study guide prepped for you. I'll get a last set of Mastering Biology questions for you. I'll let you know when I get those posted. Start thinking about that final. Make sure that you are keeping up with thinking about action potentials, and that's going into some more signaling cascades, protein signaling cascades. All right? So make sure that you're keeping up with those concepts, as well as go back and refresh your memory about all of your structures. We haven't done nucleotide structures in a while. All the amino acid structures, your translation tables. Yep. All of those things are fair game. Okay. One more, a few things about the last exam. Remember you had your last midterm exam recently. You have until Friday for point adjustments. A few students came and talked to me last week and pointed out some corrections that needed to be made. So there are a few corrections you can take to your TI now. One of those for exam version B, question 11. I had the key marked wrong. The correct answer was phosphoester, so if you were marked off for that, take that to your TI this week in recitation and get that corrected. Get those points back. Okay? So that is question 1. So that you can get a point adjustment done to your TIs at recitation. Question number 2.
Wait, wasn't it asking you how to
again for exam B, sorry, exam A. That exam didn't have any mistakes on it. For exam B,
Oh, I got that wrong anyway.
question 19.5, where you had to put down all the pathways related to what event I gave you. Because I was ambiguous in lecture about where the breakdown between phase 2 and phase 3 was in glycolysis and what was happening in each phase, or in each phase. If you got marked a point off for not having glycolysis phase 2, take that to your TI. If you just had glycolysis phase 3, I'll take that for credit. for that one point. It also has the TCA cycle for the other point. But if all you had was glycolysis phase 2 relative to glycolysis, I'll take that per point. So if you marked off for that, take that too. Yeah. Okay? As always, all other questions that are not these two corrections, or that aren't errors in addition of your points, please bring to me to talk to me about. Okay? Are there any questions on the last midterm? So be sure that you look at your exam closely if you had exam B for those errors, and make sure that you go to recitation this week and take those UTI so it can be corrected. Your exam grades, as well as your last two homework and your last two quiz grades, were posted as of last night. The average for this exam was a 71%. Okay? So go back and double check. Make sure everything's put in correctly. If there are any errors, let me know. I'll work next on updating participation points. Those haven't been done yet. Okay. Any questions about the final exam 4 corrections that I can answer for you now before we get started?
Oh wait, no.
All right, then we are going to continue talking about neuron signaling today. We're going to actually get to talk about what an action potential is. How many of you went to the dance performance this weekend called Shazam? Anyone? Did no one go? Dance performance on campus that was dance plus science? It was fantastic. They did a dance on neurons where they made the audience do the wave. And we're going to talk today about how action potentials propagate in a wave across the neuron axon. It's too bad no one was there. It was a great demonstration of neuron activity. Sorry you missed it. It's already over. But we're going to talk today about action potentials. Yeah, questions? Oh, thank you for bringing that up. So because I took, excuse me, because I took those 12 points off of exam 4, when I put it into an exam, the exam category, it was trying to force it into 125 points. So I figured it was better to leave it right now, as I didn't want everyone to freak out when they looked at their grades for that exam. So I left it outside of the exam category. So I'm going to have to go back and recalculate everything as percentage, and then repost that on La Lima so that you can get an accurate picture of what your grade is at this point. So if that is your lowest grade, it's not being calculated into your grade at this point because I've got to recalculate everything and re-enter it, which is going to take me a little bit of time. But I'm working on it. Thank you for pointing that out. So your grade right now doesn't reflect exam 4 because of that change in the total points. So I have to go back and recalculate everything, but I did want to get it up so you could see your score. Okay? So I'm working on that. La Lima is a little bit inflexible in the way it lets me do things. I discovered last night. So I'll try to get that fixed as quickly as possible so you can get an accurate picture of your grade going into the final. You can always, remember, use that weighted grade calculator webpage, or just search for one. I have a link on the announcement. You can always calculate it yourself, right? So convert all of your exam scores into their percentage, drop the lowest one, put them all together, put them in that weighted grade calculator, and that will give you an estimate. You can also use that to say, if you want your final grade to be in A, what do I need to get on the final to get there given my current score? All right? So go and play around with that as well. Any other questions before we get started? Yes? Not going to mention that today. Too many announcements already. I'll bring it up on Wednesday. Okay? All right. Any other questions? All right. Let's get started. So on Friday, we ended talking about ion concentrations inside and outside the cell relative to the neuron, and how this difference is going to set us up for generating these action potentials. In particular, we're interested in sodium and potassium. These are ions that we know have particular mechanisms of moving across the membrane. And we're going to look at how the interaction and the opening and closing of different sodium and potassium channels generate this action potential for us. But you need to keep in mind that sodium and chloride have higher concentrations outside the cell. Potassium has higher concentrations inside the cell. And this gradient is what's setting up the potential for us for the movement of ions across that membrane that's going to create the voltage we're trying to measure. Remember, this is maintained by those sodium-potassium pumps, the ATPase, which is actively pumping 3 sodium out for every 2 potassium in. So we're setting up this gradient using that ATPase, as well as just general diffusion kinetics of these 2 ions. But what we want to do now is to look at what happens now that we've set up this concentration gradient when we open up either a sodium channel or a potassium channel. And we can measure that if we measure what's going on outside the cell relative to charge and what's going on inside the cell relative to charge. All right, so I think we ended here on Monday, thinking about the potassium flow and membrane voltage. So remember, voltage is the movement of ions across the cell, and we're comparing the charge inside the cell to the charge outside the cell. All right, so if we just consider potassium channels, here's our potassium channel, here's our concentration gradient. Start off with an easy one. Which direction are potassium ions going to move when I open up this channel?
Back to the picture. It doesn't sound bad. Oh, really?
Alright, so there is the schematic again. Concentration gradient. And our channel. Which way is the potassium going to move?
I like the fruit and vegetable juice. Like, they also have carrot orange. My mom hates it. I want to pay attention.
Okay, everyone get their answer in?
Today.
Let's take a look.
Where's Louis?
Okay, so potassium concentration higher inside the cell. So therefore, it's going to move from inside to outside when we open up that channel. Remember, this is passive transport. It's following its concentration gradient. All right, so when we open up that channel, potassium moves outside the cell. They're moving positive potassium ions from inside the cell to outside the cell. So what does that mean relative to the charge inside the cell? Potassium ions are moving from inside to outside. They're positive ions. What happens to the charge inside the cell?
I mean, wait, is there no change? No, there is a change. Yeah.
Thank you.
Oh, yeah. All right. I don't get it.
All we're talking about right now is potassium. So the only thing we're opening is potassium ion channels. So that is the only thing to consider when answering this question. Okay, everyone get their answers in. Let's take a look. Okay, so we are moving positive ions out of the cell, which means relative, and we're putting them outside the cell, so that means the inside of the cell is becoming more negative. All right, so if we start at a voltage of 0, we open up those channels and measure what happens. We're going to become more negative inside the cell due to the movement of the potassium ions. So this is hyperpolarizing the membrane. Hyperpolarizing means making the inside of that cell more negative. And that membrane, we're going to hyperpolarize that membrane until there's an equilibrium of potassium ions moving in and out. So we hit that equilibrium state. For potassium ions, when channels are open, that reversal potential, also known as the equilibrium potential, so the membrane potential at which there is no net overall flow, so we're at equilibrium, things aren't moving back and forth. For potassium ions, that reversal potential is negative 75 millivolts. All right, so if that is the situation where we're looking just at potassium ions. We're looking just at the movement of potassium ions and restrict everything else. What about sodium? Let's look at what happens if we take potassium out of the picture and we look at just sodium channels. Notice again that these channels are slightly different. They can be either open and closed relative to that gate, or activated or inactivated, whether or not the drain plug is in the channel. All right, so if we open that gate, what direction does the sodium flow? Okay, everyone get their answers in. Remember where the concentrations of sodium are highest? Let's take a look. All right, because again, this is passive transport, so that sodium is moving down concentration gradient, higher concentration gradients outside the cell. So when we open those channels, sodium is going to move from outside the cell to inside the cell. All right, opposite of that is potassium. So if we're moving sodium ions across the bilayer into the cell, what happens to the inside of the cell relative to the outside? Based on the charge we are measuring. Okay, everyone get their answers in. the inside of the cell is becoming more positive. All right, we're looking at the movement of potassium and ion when we open the channels in opposite directions with opposite effects on the membrane and the voltage we're measuring. So again, if we start at that sort of same hypothetical 0 as far as voltage, and we just open up those sodium channels, the cell inside is going to become more positive. This is called depolarization. So when inside the cell becomes more positive due to movement of ions, we're depolarizing that membrane. The reversal potential or equilibrium potential for sodium is plus 55 millivolts. Okay, so when we open potassium channels, we hyperpolarize to equilibrium at negative 75 millivolts. When we open sodium channels, we depolarize to plus 55 millivolts at its equilibrium. Okay, so what is the voltage of the membrane when neither of these channels is open? Someone has done their reading. Negative 60 millivolts. We call that the resting membrane potential. All right, so if it's a negative 60 millivolts, what is moving across the membrane more in the resting potential? Potassium, because we're negative. All right. I will post some videos. You can go and look. There are some YouTube videos called 2-Minute Neuroscience, which explain membrane resting potential, action potentials in 2 minutes. They're nice little clips for review. I'll post some of those on Facebook, and they'll be in the lecture. So you should definitely take a look at those to help you go over some of these concepts. Okay, so if we're looking at these resting membrane potentials and how we're generating action potentials, we are involving different types of channels, some of which we've already talked about. So we're going to look at resting channels and voltage-gated channels. So we've talked about those different types of ion channels when we started this section and all the different ways channels can be opened and closed. We're going to look at, in action potentials, that change in voltage is due to voltage gated. So as the voltage changes across that membrane, different channels are going to open and close, versus just channels that are sort of always working at the resting membrane potential. All right, so at our resting membrane potential of 60 millivolts, we have that sodium-potassium pump, which we've already talked about. We have potassium, which has some ability to move across that membrane, diffuse across that membrane on its own. And then we have just normal potassium channels, allowing some movement of potassium, but our sodium channels are closed. So this sets up our resting membrane potential. So just to reiterate, if we measure the conditions inside the cell and outside the cell, our axon at rest, due to the movement of those ions and the way that they can leak through, and the channels that are sort of always in the background, that's negative 60 millivolts. If we look at the movement of potassium through channels only, that's negative 675 millivolts. And if we look at the movement of sodium only, that's plus 55 millivolts. All right, so that is our membrane at rest. What does that tell us about the resting membrane, the movement potential of potassium to sodium? Much more potassium. So potassium is much more permeable across that membrane. What happened So you guys have already answered that one, so I'm not going to ask you. Oh, do you want that one? Would you like that point? Okay. I'll let you have that point. Freebie. So which one is moving across the membrane the most to generate that resting potential?
I thought sodium comes in.
All right. Let's take a look.
Yeah, I guess.
Get your answer done?
Then the sodium will just become.
All right. Because we have a negative resting. Because we have a negative resting membrane potential. Someone missed that on the freebie. It's got to be potassium that's moving across that membrane more than sodium to generate that negative potential. All right. As in fact, if you look at the movement of potassium and sodium across the membrane when the membrane is at rest, sodium moves across the membrane, sort of leaks through at a rate that's 4% that of potassium. All right, so it moves across in resting state much slower, which is what generates our negative resting membrane potential. Okay. Another thing to keep in mind is that the number of ions moving across this membrane that we're measuring is minuscule compared to their concentration. So we're not talking about opening up all the channels in the cell and all the ions rush in or all the ions rush out. We're talking about opening up a few, and a few ions are allowed to move through to change that area of the membrane just enough to alter the state of our channels. Also, as we go through this, think about the way that channels can be distributed across a cell membrane in a cell. So they don't have to be evenly distributed all the way around the cell. There can be areas of concentrated ion channels and areas where there aren't many ion channels, which helps set up these action potentials. Okay, so we're going to talk about that when we talk about action potentials next. So we've already covered this a bit. Neurons are excitable. What did we say that meant? It receives a signal, and what happens? Jumps up and down in joy? Sorry. Passes on the signal how? What kind of signal is it creating? Electrical. So it has a change in that voltage in response to a signal. So we talked a little bit about what happens across the membrane. Let's put all the pieces together to look at how the neuron is generating this electrical signal that is propagated down the axon and turned into an output for the next neuron to pick up. So if we go back and look at our neuron, remember all the components are dendrites off this cell body, so multipolar neuron. The dendrites pick up a signal. When they pick up that signal, that induces a local change in the membrane to open up ion channels, in particular sodium channels. So we get a localized change in concentration of sodium. Now this localized change then spreads across the cell body passively as that sodium concentration builds up. This is helped by the fact that there aren't many sodium channels in the cell body. All right, so it has to be this passive buildup of sodium ions inside the cell body. And then once that signal has passively moved through the cell body, when it reaches that axon hillock, that transition point between the cell body and the axon, there are a lot of sodium channels here. So when that signal hits here, it can open a bunch of sodium channels at once and start that chemical cascade, opening of the ion channels, changing the membrane potential, starting the action potential, which is then going to be propagated down the axon, down here to the synaptic termini, to pass on to the next cell. Okay, so when that signal hits the axon hillock, what is actually happening to the cell membrane at that point? So let's go back to an axon at rest. Our axon is just chilling, hanging out, having a good time. What happens when it gets a signal or there is a change inside the cell to increasing sodium? All right, so if we give it a little bit of a signal, we see that basically it just tails off, right? There's a little bit of an input of charge, change in charge, and then the membrane slowly comes back to resting potential. What happens if we give it a larger signal? All right. So the question is whether or not this cell is excitable. So if we give it a signal, nothing happens. We give it a small change, nothing happens. Give it a small change, nothing happens. Give it a slightly larger change. We generate a response. So is this cell excitable? Think about what it means to be excitable. For a cell, anyway. What did we see happen in this cell? What is the definition of excitable? Are we jammed or is everybody still thinking? We're stuck, aren't we? All right. Take that one off. Good thing I gave you that CD earlier. I'll make up for this one. All right. So is this cell excitable? Yes. We gave it a stimulus, and there was a response in terms of the change in the voltage across that membrane. So what are the properties of an excitable cell? Generates an action potential in response to a signal. Do all stimuli excite an excitable cell? See if this one works. Must be the heat making my receiver break down. No, that bill stuff. Okay. Well, we'll give it a shot. All right, so as I showed you in the last slide, to be an excitable cell, not all stimuli are going to generate that action potential. Right? In order to generate an action potential, you have to hit some threshold value to set off that response. So small changes, this keeps the cell from constantly firing to every little bump that it gets, right? So you have to have a stimulus that's going to change past the threshold value, and then you get that action potential. The action potential being the change in that local membrane potential, first depolarizing, then hyperpolarizing, and then back to resting membrane potential. The next question, does the response of the cell vary with the stimulus? So if I give a larger stimulus, do I get a larger response? Yes, no, maybe, there you go. That's all possible answers. Depends on the nerve. So let's take a look. Get this out of the way. All right, so if we have our cell membrane resting potential, we give it a stimulus past our threshold, which in this case is negative 40 millivolts. We generate that action potential, goes back to resting membrane potential. We give it a larger response, same action potential. So the response doesn't vary with the stimulus if it's above the threshold. This is because we're talking about a localized region of membrane and the set of ion channels that are in that localized region of membrane. When you get past the threshold, you open them up. It's not a graded threshold where a little bit more opens up more channels, right? When you get past the threshold, the channels open up and you get a response. Okay. So the response of the cell does not vary with the stimulus, as long as that stimulus is above the threshold. Below the threshold, you don't get a response. All right, so we're working actually looking at how these action potentials are generated. Let's look at what happens in the resting membrane potential. Now we're going to get to our voltage-gated channels that are generating these responses, these electrical responses due to stimuli in the cell. Okay, so let's look at sodium and potassium. We looked at just sodium channels and just potassium channels, so you ought to be able to roughly guess which channel is responsible for which part of our action potential, the action potential being this response. Right. This way is depolarizing, this way is hyperpolarizing. Remember which channels lead to those membrane responses? Excuse me. So if we look at sodium channels, the ones involved in generating these action potentials, they are voltage gated, meaning that they are going to change their state relative to the membrane potential. So if we start at our resting membrane potential, negative 60 millivolts, our sodium gated, our voltage gated sodium channels are closed, but they're activated because that ball and chain is not plugging the channel. When we hit that threshold value for the stimulus to change the membrane potential to negative 40 millivolts or higher, that induces the sodium channel to open. So when the sodium channel is open, sodium comes into the cell, the inside of the cell becomes more positive. We depolarize. All right? We hit that threshold, that induces those sodium channels to open up the gate so that sodium ions can move through, depolarizing that membrane, leading to this part of our action potential. So let's finish up the cycle for the sodium channels. So as the cell membrane is depolarized, so the threshold is at -40, which opens up the channel, sodium comes in, the cell membrane is depolarized. When it gets to +40 millivolts, our sodium channel is inactivated. We see another conformational change in this channel. The ball and chain plugs the channel and keeps sodium from moving across. And then as that membrane potential drops, when it hits -65 millivolts, we again see a conformational change in this ion channel back to its original state. So the gate closes and the ball and chain drops out, so it is now closed but activated and ready to go again and receive a new stimulus. Okay, so voltage gated sodium channels. This is how they work. They open at that threshold value. This is what's starting our action potential. So when the membrane potential changes to about -40 millivolts, we open up the gate on our sodium channels. They inactivate after about a millisecond due to the depolarization of that membrane. And then as the membrane hyperpolarizes, so it comes back negative, it de-inactivates at about -65 millivolts, which resets that protein to receive a new stimulus. Notice that it cannot reopen until you de-inactivate it. So you get down to -65 millivolts, and our ball and chain drops out. All right, so once it is inactivated, this sodium channel can't be turned back on until it's de-inactivated, so it's dropped down to that membrane potential. All right, so sodium channels are what creates the first part of our action potential, that rise, that depolarization of the membrane. Why does the membrane voltage drop? What causes that membrane voltage to drop, to hyperpolarize? Our potassium channels. So our potassium channels get involved at that point. So the kinetics of opening of sodium and potassium channels is different. Sodium channels open quickly. Potassium channels open up slowly. And so as the sodium channel is sort of closing, the potassium channels are finally getting around to opening, which switches the membrane from depolarization to hyperpolarization, bringing it back down towards resting membrane potential. All right, so there's another 2-minute neuroscience video that you can take a look at. I'll post on Facebook for you. With the last few minutes of class, let's just put this all together. So let's look at our sodium and potassium channels together. We're at our resting membrane potential, -60 millivolts. We hit our threshold value of -40 millivolts. Potassium channel opens up. Sodium moves into the cell. That membrane potential depolarizes up to about 40 millivolts. That inactivates the sodium channel. About this time, the potassium channels wake up and open up, allowing potassium then to move across the cell in the other direction. That's going to drop our membrane potential, so we're hyperpolarizing. Notice that we overshoot resting membrane potential by a little bit. which resets our sodium channel. And then at that point, our potassium channel starts closed, and that membrane is ready to receive a new stimulant. So we have our resting state, our depolarizing phase, due to the opening of sodium channels, our repolarizing phase, or hyperpolarizing, due to the opening of potassium channels, our undershoot, so we actually go past our resting membrane potential, which resets our sodium channels, and then we slowly come back to resting state. All right, let's end there. We will finish up, I think, after potentials on Wednesday. We have homework due. All about